The last bit that we have to discuss in Chapter 8 are enzymes. Enzymes are catalysts, which are things that lower the activation energy needed for a chemical reaction to occur. In other words, they make something require less energy to get the desired outcome. Enzymes are the catalysts found in living organisms. The example that we're looking at on the screen here, we see sucrose being broken down by sucrase, the enzyme, into two smaller molecules. You'll notice here sucrase ends in ASE. Most enzymes will end in ASE. So if you ever see a word and ends in ACE, it's probably talking about an enzyme of some sort. A couple of other items worth noting here. First off, you'll notice sucrase is awfully close to sucrose. And the reason that it's named sucrase is because enzymes are specific. Sucrase will not break down any other thing in the body. It is specific to sucrose, this particular sugar, and it will not act on something else. Another thing I wanted to point out once again, water is being added in this reaction, so it is a form of hydrolysis. So looking at activation energy, or EA here, activation energy, we can see a couple things happening. First off, chemical reaction. We have reactants on one side, products being formed on the other side. We can see that the reactants over here have a higher free energy than the products over here, which means that energy has been released, so we have a negative delta G here. And we can see that energy had to go up in order for that energy to be released. So we had to have some energy put into the system. Every chemical reaction between molecules involves bond breaking and bond forming. The initial energy needed to start a chemical reaction is called the free energy of activation, or simply put, activation energy, EA. Activation energy is often supplied in the form of heat from the surroundings. Enzymes catalyze reactions by lowering the activation energy barrier. With enough activation energy, the bonds of the reactants break, resulting in an instability in the reactants. This stage, what we see at the top of the graph here, is called the transition state. And we can see the bonds kind of being represented as not stable, which is the wavy line that we see going on there. The enzymes do not affect the change in free energy, or the delta G. Instead, they accelerate reactions that would occur eventually. As previously mentioned, enzymes are specific to what they act on. What an enzyme acts on is called the substrate. And substrates bond to an enzyme in what is called the active site, which is right here in this case. Once a substrate has bonded to an active site, the enzyme will kind of surround it to act upon it, and at this point we have what is called an enzyme-substrate complex. To perhaps give you a better idea of what's meant by enzyme specificity, picture a Phillips screwdriver and a flathead screw. If you try to use the Phillips screwdriver on the flathead screw, it's not going to work out so well. Similarly, tire iron is meant for the lug nuts on a tire, and they fit perfectly because that's what they're designed to do. That's exactly how enzymes work. They have one substrate that they act on, and the enzymes seek out those substrates and they wait for them to bond to the active site, really before they do anything. In the sample we see here on the screen, we have an enzyme here, and we have the little dip there and the little crevasse up here, and we have a couple substrates that are going to go into that active site kind of together. And in order to form this enzyme substrate complex, the enzyme itself has to orientate these substrates to kind of fit exactly what's needed, kind of like putting a puzzle in the right spot here. And when this orientation is being done, that's actually called inducing fit. So induced fit is taking substrates and arranging them in a specific orientation within the active site of the enzyme. Once the substrates are in the active site, the activation barrier can be lowered by orienting the substrates correctly, straining substrate bonds, providing a favorable microenvironment and covalently bonding to the substrate itself. Notice in our cycle here, it is a cycle, that an enzyme accepts substrates, orients them, speeds up the process in changing these substrates into something different. They're changed into products. Once that's done, the enzyme releases them, and the enzyme is ready for more substrates. Looking at the optimal conditions for an enzyme to function, it's fairly logical. I mean, human bodies are optimized at certain temperatures. Think about if you go out into the middle of winter and you're standing on a lake and you're freezing. Are you going to be ready to run at your fastest speed in those cold conditions? Well, no, because your muscles are all tensed and tightened up. You can't function at that temperature, not at your highest ability anyways. So enzymes really are no different than that. Temperature, pH, are a couple things that affect the functionality of an enzyme. Each enzyme does have an optimal temperature in which it can function, and each enzyme also has an optimal pH in which it can function. For the enzymes that occur for digestion in our stomach, they have to be optimized for a pH of 2. And actually what we're looking at here on the screen, pepsin, which breaks down proteins, is in the stomach, and the pH of the stomach is 2, so it makes perfect sense that its optimal pH is right here at 2. A little bit too acidic, it still functions, but not optimally. When it's a little bit more basic, still acidic, I guess, looking at the pH there, 3, it doesn't function as optimally it does as a pH of 2. Next, we have non-protein enzyme helpers, known as cofactors. Cofactors may be inorganic. An example would be a metal in an ionic form, or they could be organic. An organic cofactor are vitamins, and those are called coenzymes. Enzymes that require cofactors or coenzymes are inactive without the substrate. So they're just there, they're ready for the substrate, but they don't do anything unless the substrate is present. Notice the specific bonding site and substrate shapes that we see going on in the image here. Substrate cannot fit in without the coenzyme fitting in first to give it the unique shape for the active bonding site. There are also enzyme inhibitors. Two types that we're going to look at. We're going to look at competitive inhibitors and non-competitive inhibitors. To start with, the competitive inhibitors bind to the active site of the enzyme, so the substrate actually has to compete with it. We see that going on right here. The competitive inhibitor binds to the active site, substrate can't, substrate cannot be made into a product. The non-competitive inhibitors are inhibitors that bind to a different part of the enzyme, ultimately causing the enzyme to change shape and have a less active bonding site for the substrate. And image C here shows that. We have our non-competitive inhibitor locking into the bottom of the enzyme, causing the active site itself to slightly change shape right here. And that reduces the ability of the substrate to bond into that active site. So what about if enzymes are present but aren't necessarily needed? Well, the text stated that chemical chaos would result if these metabolic pathways were not tightly regulated. A cell does this by switching on or off the genes that encode for specific enzymes, or by regulating the activity of the enzymes. What I mean by turning on or off the genes is this. Your body is capable of taking a section of your DNA and turning it off just like a light switch, rendering it inactive. However, there are times when that gene needs to be used, such as to produce a certain enzyme for a specific function, and the light switch goes on, and the DNA starts producing the enzymes needed for whatever situation might be required. For the enzymes that do not necessarily need to be remade, they're just always there, those are controlled by allosteric regulation. So the allosteric regulation may either inhibit or stimulate an enzyme's activity, just depending on the needs of the cell at the particular given time. So our top few images up here show us allosteric regulation, one with an activator, one with an inhibitor. You can really almost think of this kind of like I don't know, a baby stroller and the little locks that you can push down the wheels so that the wheels don't turn whatsoever, or flipping them up and they roll freely. So with the activator, it allows the active sites to remain active, do their job, break down substrates. But put the locks on and the wheel stops rolling, bonding sites close, and nothing can be done. It just kind of waits around for it to be activated again. Now, this is actually the bottom part of the last slide, cooperativity. Enzymes are in their inactive form, and sometimes it just takes one substrate to kind of get the ball rolling. So we have these kind of closed or sort of muted active sites, but if a substrate can bond into one of them, it makes the other ones a little bit more ready to kind of do their job as well, kind of a snowball effect. So this is called cooperativity, where one substrate can bond in, and it actually enhances the ability of the enzyme's other active sites to do their job, provided the enzyme has multiple active sites. And lastly, we have feedback inhibition. Feedback inhibition, the end product of the metabolic pathway shuts down the entire pathway. Feedback inhibition prevents a cell from wasting chemical resources by synthesizing more product than is needed. This image illustrates the conversion of threonine to isoleucine, which are two amino acids. When necessary amounts of isoleucine is produced, the initial enzyme that started the process is inactivated. So we see the threonine going through a series of reactions here, producing isoleucine, and once enough isoleucine is produced, the isoleucine itself acts as an inhibitor to the enzymes, shutting them off, and then once the isoleucine gets used up, we have a functional enzyme again ready to continue breaking down more of the threonine. That concludes Chapter 8, Metabolism. If you have questions, please bring them to class with you or shoot me an email so that I can look into those for you.